Ford, who will be your host over the next 2 days. And now I'm sure you're ready for us to launch fully into today's agenda. And as a reminder, all our speaker bios are available on the One Stop Shop website. But for now, I am honored to introduce Dr. Anil Patri, who will officially welcome us all. Dr. Anil Patri leads FDA's Nanotechnology Task Force and oversees collaborative regulatory science research at the Nanocore. His lab developed 7 international standards in nanotechnology through ASTM International in the past 4 years and contributed to many other documentary standards. Dr. Patrice serves on the Nanoscale Science, Engineering, and Technology Subcommittee on behalf of FDA for interagency coordination and leads the US EU communities of research and characterization. Prior to joining FDA in 2014, Dr. Patrice served as the Deputy Director of the Nanotechnology Characterization Laboratory at the Frederick National Laboratories for Cancer Research and as a guest scientist at the National Institute of Standards and Technology. Let us please welcome Dr. Anil Patri.
Thank you very much, Brenda. I echo your comments about the many that contributed to this symposium. Good morning, everyone, and thank you for joining us today. My name is Anil Patri, and I would like to welcome you all for this year's FDA NanoDay Symposium on continuous manufacturing of nanomaterials. I would like to thank Brenda Stoddart and CDER SBIA program for assisting us with the NanoDay Symposium every year. Some of you may have attended the last year's symposium, which is highly successful. And for this year, especially, I thank Dr. Xiaoming Xu for leading this year's symposium. It continues to be an exciting time for nanotechnology with last week's 2 Nobel Prize nods for research on mRNA COVID vaccines for physiology and medicine, and for research and application of quantum dots in chemistry. Today's symposium is dedicated to the emerging continuous manufacturing of nanomaterials intended for medical products. Continuous manufacturing is in use for small molecule drugs for some time and only recently applied to the field of nanomaterial synthesis, especially for liposomes and lipid nanoparticles, and can be extended to others. FDA has funded this effort through broad agency announcement grants, and we have an excellent panel of speakers today, both from within FDA and those that are funded by the FDA, to present about the past, present, and future applications, and how this will potentially address challenges in drug shortages while assuring the quality products to benefit patients. I would like to now request Dr. Nemanje Bumpus, FDA Chief Scientist, to provide opening remarks. Dr. Bumpus joined FDA a little over a year ago with distinguished career in academia, more recently at Johns Hopkins University as a professor and chair of pharmacology and molecular science department. At FDA, she is spearheading the research, science, and innovation that are fundamental for our regulatory mission. Dr. Bumpus?
Welcome, everyone. I am Nemanje Bumpus, the FDA Chief Scientist. I would like to congratulate the Nanotechnology Task Force and the CDER Small Business Industry Assistance Program for organizing this NanoDay Symposium to showcase FDA's work on continuous manufacturing of nanomaterials. I would also like to thank them for the excellent work that they do throughout the year. The mission of the Nanotechnology Task Force is to leverage FDA scientific expertise and resources to advise agency and center leadership on critical and cross-cutting nanotechnology-related issues to support the development of safe and effective FDA-regulated products that may contain nanomaterials or otherwise involve the application of nanotechnology. This symposium serves that mission. FDA has been more actively involved in celebrating NanoDay over the last 4 years. In 2020, We celebrated it by releasing the Nanotechnology Task Force report. In 2021, we had an internal research symposium at FDA for NanoDay. In 2022, we had a public meeting to highlight the final guidance to industry from CDER and CDER on nanotechnology and drug products. Last year was very successful with over 3,500 virtual participants. This year, we wanted to highlight the emerging area of continuous manufacturing due to its importance in advanced manufacturing, a topic of interest within FDA and globally. Continuous manufacturing is an emerging technology that improves product quality, reduces waste, lowers production costs, and enhances manufacturing flexibility and agility. This symposium intends to share FDA's experience in this emerging area and hopes to inspire developers and researchers to adopt and realize the full potential of nanotechnology and continuous manufacturing. We're only starting the journey. From a research standpoint, significant work lies ahead to enable development and translation of therapeutic and vaccine products utilizing continuous manufacturing. Enjoy the symposium and happy Nano Day!
I would like to now introduce Xiaoming Xu, the Division Director for Office of Testing and Research, Office of the Office of Pharmaceutical Quality, CDER, FDA. Xiaoming also serves on the Nanotechnology Task Force as a member from CDER along with Olin. And he has been very active in both research at FDA in nanotechnology and leads an excellent research group. So he's the main organizer for this symposium. So I would like Xiaoming to say a few words before taking it over. Xiaoming?
Hello, everyone. I'm Xiaoming Xu, the co-chair of the 2023 FDA NanoDay Symposium. I want to start by recognizing my colleague, Dr. Anil Patri and Dr. Olin Stevens, for their guidance and help over the last few months planning for this event. Together, also, we would like to thank our amazing staff at SBIA for organizing today's event. In the next few minutes, I want to briefly share with you our thought behind this event and introduce to you the topics that will be covered. This symposium is focused on the convergence of two interesting fields, nanotechnology and advanced manufacturing. It's a rapidly developing topic, particularly at the intercept of the two fields. As you may know, to date, we don't have yet any regulatory approval of nanotechnology products manufactured by continuous manufacturing. However, we hope that you can see the potential, maybe after today's event, and that we can all work together to push forward the field, and maybe one day to approve the first nanotechnology drug product manufactured by continuous manufacturing. Now, as you might also notice, the title of this slide includes the phrase, learning from the future. Isn't that strange, as we typically only look into the past to learn? Let me briefly explain it. As we have all experienced it, we tend to conceive time as linear and unidirectional, such as moving from the past to the present and to the future. We remember yesterday, we experience today, and we look forward to tomorrow. However, to plan or to prepare for the future, especially in uncertain times, sometimes we need to embrace a nonlinear conception of time. And this concept is coined by Dr. Peter Skoplik in his 2020 Harvard Business article named Learning from the Future. What this concept entails is that sometimes we need to step away from the traditional view of time and sequence of events to take a leap forward as well as backwards so that we can influence the future instead of just anticipating it. As he described in the article, sometimes we need to take a closer look of the trends in the present, then jump years into the future and to describe a plausible world of future scenarios, then work backward to develop stories about how the future world may become, and then work forward again to develop strategies to realize that future. And this concept is widely used in the business field. Also, it's called strategic foresight, which is drawn from many successful cases from other industries. It prepares the organization to better weather the storm in particularly difficult times, which is often hard to predict, such as a COVID-19 pandemic. So I bring up this concept as a preface to the conference because I see parallels in scientific research. Maybe this view of the connection is easier to understand. What we view as today's success, especially in scientific breakthrough, is often a deliberate effort from the past. After all, scientific research takes long time and dedication to accomplish. For example, concept of nanotechnology is not new. Indeed, Dr. Benham identified the spontaneous formation of vesicles from phospholipids more than 6 decades ago. And since then, countless researchers had dreamed big and started experimenting with lipid vesicles to encapsulate various types of therapeutic agents to take advantage of this natural delivery platform. And you must have heard about the news last week of the Nobel Prize Award to Dr. Kathleen Carrico and Dr. Drew Weissman for their contributions to the messenger RNA research, which is a crucial tool in combating the COVID-19 pandemic. Big congratulations to them for such worthy recognition. If we think about it, the anticipation of the use of filtrogenic lipids to help with the delivery of RNAs has been perceived more than 2 decades ago. And today's success in COVID vaccine did not begin after the pandemic, but instead started much earlier. So, as we celebrate today's success, we cannot overlook the effort from the past, which was often a deliberate effort to change the future. Speaking of nanotechnology, I would like to remind everyone about the view at the agency about if and when do we consider FDA regulated product utilized nanotechnology. The first point to consider emphasizes on the dimension of the engineered nanomaterials, while the second point to consider emphasizes on the purpose and impact on properties relevant for products' intended effect. Evidently, over half of FDA-approved drug products containing nanomaterials with dimension between 100 nanometers and 1000 nanometers. So when you hear the term nanomaterials, do not assume it is only for those that are less than 100 nanometers. Now let's go back to the theme about learning from the future. What we can imagine as the future that we can work towards today, especially in regulatory science research. Maybe it's making the nanotechnology product more accessible, even more robust against drug shortages. After all, many patients cannot afford to lose access to their life-saving drugs, such as oncology treatment or crucial vaccines. Maybe it's to prepare the industry and the regulatory agencies to be more agile so that we can more easily introduce or tolerate changes, whether it's to change a supplier of a raw material or changing location of the manufacturing. Maybe it's to better prepare for a future of more individualized treatment. For example, with so much progress made in the human genomics and understanding of individual risk factors, can we develop products targeting individual patients rather than a one-size-fits-all approach? Of course, we desire a future where the nanomaterial drug product can become more affordable, so we need to bring forward paths to encourage the development of the generics. Lastly, patients need to trust that industry has developed a medicine that has been rigorously evaluated so that they have confidence about the use. Now, let's all think about a future where it is better than today and how we can get there. Speaking of trust, everyone deserves the confidence in their medicine, not just the one went through the clinical trials and approved by the FDA yesterday, but the ones that the patient will receive tomorrow in their hand. What allows them to have the confidence is the pharmaceutical quality, which ensures that the availability, safety, and efficacy of every dose is something we need to continue to improve. One way to improve the quality of drug product, and certainly it is an opportunity area for nanomaterials, is the use of continuous manufacturing. Similar to nanotechnology, continuous manufacturing is not a new concept. However, in pharmaceutical applications, it is a rather new application. We have one talk immediately following this one to provide a more detailed explanation and overview about FDA's experience so far on continuous manufacturing and what we can learn to apply to nanomaterials. To close my portion of the talk, I would like to introduce to you the presentation topics of today. You will first learn about our experience in developing and approving a continuous manufacturing drug product, kind of what is a current trend. And then we will share with you our past effort invested in the area of continuous manufacturing of nanomaterials from 2 FDA grant awardees at the University of Connecticut and Princeton as well as Purdue. We will follow that with some discussion about current regulatory challenges and opportunities, as well as ways that you can interact with the agency to help pushing forward the regulatory science research. Lastly, let's start taking action today so that we can all change tomorrow. And thank you very much, and I hope you will enjoy this symposium.
Thank you, Dr. Xu, for setting the stage for the remaining presentations. This is Ray Ford working with SBIA, and it's my pleasure to be your host for the remaining sessions. Moving into our first presentation on CDER's perspective on continuous manufacturing journey, past, present, and future. This will be provided by Dr. Thomas O'Connor, the Deputy Office Director in the Office of Testing and Research in OPQ. Next, we'll learn about continuous manufacturing platforms for lipid and polymer-based nanoparticle therapeutics from Dr. Antonio Costa, University of Connecticut. For a complete biography on each presenter today, please refer to our speaker biographies located on the event page for all event details adjacent to the event agenda. Please join me to welcome our first presenter, Dr. O'Connor.
Hello, my name is Thomas O'Connor, and I'm excited to join you today to talk about CDER's perspective on the continuous manufacturing journey. First off, I want to focus the talk on pharmaceutical quality. So that is really that every patient deserves confidence in their next dose of medicine. Pharmaceutical quality is really assuring the availability, safety, and efficacy of every dose. I want to start off today's talk by defining what do we mean by continuous manufacturing. Certainly, each individual unit operation in a manufacturing process could be continuous, where the material continuously flows in and flows out of the unit operation. But when we're talking about continuous manufacturing, really focus on an integrated process that really consists of 2 or more unit operations that are connected together. And what that means is that there's less isolated material during the course of the processing because of this integration, and that drives more reliance on real-time data collection and decision making. And FDA has identified, long identified, continuous manufacturing as a novel technology in the pharmaceutical industry, which may not be a fit for every drug, but can bring potential benefits related to increased efficiency. It usually results in more intensified processes that reduce the footprint of the process. And again, this increased data collection in real time to facilitate the control of the process improves the process control. And while at the start of this journey, those benefits were potential, we have illustrated examples where the companies have realized those benefits. So this is a report from one company that switched from a batch process to a CM process and reported significant reductions in operating costs and reductions in efficiency, so reducing their waste, and increasing both their cycle time for manufacturing as well as testing. Using more of that data from the actual process to speed up some of the end product testing. And not only does that translate to benefits that can be quantified in that respect, CM can also enable processes that just couldn't be safely conducted, using batch manufacturing processes. This is one example reported by a company that used CM for a Grignard reaction, which results in allowed spontaneous heat release. So the continuous process was able to be safe and well controlled and really enable this reaction to take place, which couldn't be safely executed in a batch reaction mode. From the FDA's perspective, not only are there potential benefits, for industry, but there are also potential benefits for patients, including, better quality medicine. This improved control can reduce, the amount of defects that are produced. Continuous manufacturing, usually involves less scale up because it's intensified processes, resultant processes that can be. You can kind of use the same rig for development as you do for commercial manufacturing because they're smaller and use less material, and that can really help speed the development and getting those products to market faster, especially if for these drugs that have accelerated clinical development timelines. By reducing quality issues can help prevent drug shortages. And then having more flexibility and agility in the manufacturing sector can help improve emergency preparedness. So having a manufacturer that can quickly scale up the amount of product being manufactured. So I just want to kind of take you through a little bit of a timeline to review some of the key highlights along this continuous manufacturing journey. It's labeled here, and I'm going to start off in 2014. Not because 2014 is actually the beginning of the journey. That actually started much earlier when there was a number of investment in some of the science and actually the equipment that happened both in academia and industry that predates that by over the previous decade. But 2014 is really kind of a turning point, a tipping point, as you will. So at that year, we had our first international symposium in continuous manufacturing, really bringing together international regulators together for the first time. And we had the forming of CDER's Emerging Technology Program, which has really been the program that's really interfaced with companies looking to implement continuous manufacturing. 2015, big milestone, the first drug product approval that was made using continuous manufacturing, as well as two papers that published that really outlined some of the scientific thinking behind quality considerations. One was a joint industry regulator paper, and the other paper was from the FDA. In 2016, we even had the first approval of a product that was made by batch manufacturing, switching to a continuous manufacturing process. In 2017, we continue to see additional approvals in the drug product space, as well as finalizing the guidance for the emerging technology team, a key milestone for that program. 2018, we see the formation of ICH Q13, which is an international harmonized guidance focused on continuous manufacturing, so endorsement. So that's just the beginning of that process, but there's a key milestone leading to international harmonization. 2019, again, you see additional approval, but also FDA publishes draft guidance on quality considerations for continuous manufacturing. It's important to note that that guidance is going to be more limited in scope. It's going to be focused on drug product and solid oral dosage forms, which really form the basis of our experience up to that point. In 2020, we start seeing approvals in more expanded applications. So we have first approval for a continuous drug substance process, first approval for a semi-continuous biotechnology process, even for sterile drug products, these semi-continuous processes. So we're starting to see continuous manufacturing being adopted by for other types of drug products. And then 2021, a milestone was reached, which is the graduation of the continuous direct compression from the Emerging Technology Program. That really signifies that we had enough, knowledge gained over the past, 7 years that those type of applications can actually move from, the Emerging Technology Program and can be really handled, through the normal assessment channels. That industry had enough experience implementing these type of processes. 22, first approval of a CM process at a CMO facility. So not only these processes, commercially available at innovator companies, but actually made available to others at a CMO complex. In 2023, brings us up to the present day. We have another just getting more complex products being made by continuous manufacturing. This one, a peptide product. And then you also see the publication of ICHQ13, which is, again, a much broader scope than that original FDA guidance, because it covers not only drug products, but also drug substances, and it's not limited by type of modality. And we'll discuss this more later in the presentation. So as we went through that continuous manufacturing journey, CDER had basically 3 types of programs that really supported us getting to where we are today in that journey. So one is focused on assessment inspection. That's the emerging technology program. But we also had key, in the science area. So investing in advanced manufacturing science and research was really important. And of course, taking those learnings both from assessment and from the research, and really helping us develop policy. And this led to the publication of ICHQ13, which is actually a harmonized guidance for sponsors looking to implement continuous manufacturing. So I'll talk, for the rest of the presentation, talk a little about each of these different programs and how it relates to where we are in our continuous manufacturing journey. So the first program I'll talk to you about is the Emerging Technology Program. So this program has been around since 2014, as I mentioned. So many of you in the audience may be very familiar with this program, but for those who have not interacted with this program before, it's really, provides, it's really focused on promoting and facilitating adoption of innovative approaches to pharmaceutical product design and manufacturing. It's a cross-functional team. So today we have about 30 members, and it can always pull in additional ad hoc subject matter experts, and they represent all the relevant FDA quality review and inspection programs, including the Office of Pharmaceutical Quality, but also the Office of Compliance and the Office of Regulatory Affairs, really providing that one quality voice. And the program works by providing an opportunity for industry to engage and collaborate early with the FDA, discuss, identify, resolve technical and regulatory issues. And this early engagement can even before a specific product is identified. If you're considering an emerging technology that could be used across many different products, you can still approach the emerging technology program to discuss some of your early technical and regulatory questions. So the objectives of the program really kind of first of all serves as that centralized location for external inquiries from stakeholders, and really provides that forum for early engagement and dialogue, again, even if a particular product hasn't been yet identified. And from that early engagement, the program is designed to stick with that program throughout its maturing into eventually application, hopefully approval, and commercial production of a product. And that helps ensure the consistency and continuity and predictability in the review and inspection of that. Again, through that early engagement and all the way throughout that life cycle is identify and evaluate roadblocks, right? Whether related to existing policy or practice. And then as technologies mature, really to facilitate that knowledge transfer and help establish scientific standards and policy as needed. The ET Emerging Technology Program is really focused on a collaborative approach. That means internally, in the FDA, collaborating, forming those cross-collaboration teams, as well as our engagement with industry. So it starts with our early engagement. Another unique aspect of the program is the emerging technology site visits. So these are non-inspection visits, but it gives us a chance to go out to the manufacturing facility where the continuous manufacturing or other innovative technology is being implemented and have more detailed discussions. I can also engage in discussions focused on the pharmaceutical quality system, on readiness for inspection, and those kind of questions. Make sure the eventual application is as accessible as possible. But it continues on all the way through that when the application arrives, the integrated quality assessment. The ETP is serving as a co-lead of that application assessment and can participate in the pre-approval inspection, along with investigators from ORA. And the ATP has had many engagements related around continuous manufacturing. In fact, it's probably the technology we've had the most engagement with, through the program. And through those engagements, we've kind of identified some of these control strategy considerations. So, one of that is that, now you have material flowing through the process. And so as you need to reevaluate some of the raw material specifications, right? Because there's new requirements for that material. The process is dynamic, and so we need to characterize those process dynamics. Typically it's done by characterizing the residence time distribution and understanding how the system's going to respond to if it's disturbed during the process. The process monitoring control strategy often involves process analytical technology, so that's usually a key discussion point. Increasingly, we're seeing models being used as part of the control strategy, again to help us monitor and ensure product quality. Material collection and diversion. Because product is not being collected all at the end of the process, it's being collected over time. If there is an issue during the process, that material can be collected and diverted from the good product. There are known instances when this might be happen, such as startup and shutdown, but there could be unforeseen issues that might trigger this, and so there needs to be strategies to identify, isolate, and divert those materials if those do happen. Often, and also there's the potential for real-time release testing, and we see many implementations of that for continuous manufacturing. So not only are there control strategy considerations, but there's considerations for the facility and quality management system. So there might be need to updates to the quality and production procedures. Often continuous manufacturing involves more automation, more collection of more process data, more process analytical technology, and so there needs to be sorting out of quality's oversight of those elements. Again, the equipment training is integrated, so not only you're performing your qualification for individual pieces of equipment, but you need to focus on the qualification of the entire train. And if that equipment is being used for multiple products, there's things like cleaning and maintenance that needs to be procedures for those need to be developed. Process validation. So we see a lot of questions around this because a lot of the data developed during development, because it's being used to use the same process for continuous manufacturing, can actually give you knowledge that could be used to support process validation. So there's kind of a blurring a little bit of the individual steps for the process validation, but at the end of the day, you still need to demonstrate a robust process monitoring and control strategy. And again, if you do implement material diversion, what is the quality's role going to be in investigating those events? That needs to be kind of determined ahead of time. Advanced manufacturing research. So it's really important, as we talked about all those elements where the Emerging Technology Program has interacted with industry. It's really important the feedback on those points really be informed by the state-of-art manufacturing science, and that's really the goal of our research program. And it's really a dialogue with the Emerging Technology Program where questions could be raised to industry. They're looking for ETT feedback, and when the answers to those questions are maybe not clear, or we need additional data, that's an opportunity for research to kind of support enhancing our understanding of that particular issue. And that research can be conducted intramurally within the FDA, or it can be conducted through a collaboration with academia or an outside partner. But really the end goal is really to have shared learning and communications, both from the industry side and the FDA side as those technologies get developed and are being implemented. And for advanced manufacturing, there's a number of different research areas our research program is focused on, including novel manufacturing operations. Sometimes switching to continuous doesn't mean just connecting pieces of equipment together. There might actually be new manufacturing operations. Again, we've looked at advanced manufacturing for complex products, so that brings an additional complexity to that, as well as ways to monitor control our processes, whether that's process analytical technology or process modeling. And the goal for these programs or these research projects is to really share the information that's generated from them. So over 100 publications have been produced from these projects to contribute to the community's knowledge. But that's not the only impact the research has had, because it has really directly informed our regulatory feedback and decision making. So it's either providing recommendations to the emerging technology program, really informing that, or informing the actual assessment of regulatory submissions that included continuous manufacturing. In fact, almost most of the submissions that have been approved by the FDA have been impacted by the FDA's research program. And also inform policy development, particularly in areas where we may not have a lot of regulatory experience yet. And that can be including elements of ICH Q13 or ICH Q5A, which is the guideline for viral safety. And when you're talking about continuous manufacturing for biotechnology products, that is a key potential issue. CDER is, just to call out one example, CDER has done a lot of research on process models, and building those models has been actually applied to aid the assessment of a number of continuous manufacturing applications, supporting approval of those applications. So that work has also been recognized externally. Finally, I want to talk about, from the policy perspective, ICH Q13, which is a guideline on continuous manufacturing of drug substance, drug products. So you can see there's a challenge when developing ICH Q13, giving it broad scope, right? Is how to address all these considerations for different types of processes, different types of modalities, and really to tackle that, they came up with a strategy where they develop a main body of the guideline that covers fundamental aspects of continuous manufacturing that are not going to be specific to the technology or dosage form. So these contain some basic CM concepts, some scientific approaches, and regulatory considerations. Then they developed annexes that can provide illustrated examples and provide considerations specific maybe to a certain modality or production method. So in the CM concepts, it talks about defining batch definition, because unlike batch manufacturing, continuous manufacturing, batch size is not necessarily constrained by the equipment size. And there can be flexible approaches, based on the quantity of output material or quantity of input material or runtime at a defined mass can be used to define the batch. And there's different modes of continuous manufacturing, right? We know that we've defined it as 2 or more integrated operations, but that can really be a partial, part of a process, or it can be the full, equipment train for a drug substance, drug product process, or it can even bridge, that drug substance, drug product process. For scientific approaches, it really focuses on this holistic approach to understanding process dynamics and designing the control strategy. Highlights the potential to use material diversion as part of the control strategy, which is unique to continuous manufacturing. It talks about the scientific considerations for changes in production output, whether you're changing the runtime or the mass flow rate, or scaling up the equipment. Each of those has different scientific considerations. It also identifies the potential of using the data generated during the process to really support continuous process verification as an ongoing way to verify the performance and the product quality. It also then talks about some regulatory considerations, clarifying where in the application certain continuous manufacturing specific information should be included, describing aspects of the pharmaceutical quality system with respect to managing disturbances and material diversion, highlights options to generate batches for primary stability data. It can be a single manufacturing run versus multiple shorter runs. There's different ways to kind of generate that key data. So finally, I would just like to conclude my talk with some concluding statements. So when we look at the totality of these 3 programs, from the Emerging Technology Program, research and policy, what has the impact of that been? And so we did a self-audit that looked at approved continuous manufacturing applications for solid oral drug products, which is where the most of our experience has been to date. And what we found is that actually, these applications actually had shorter times to approval and faster times to getting to the market than a cohort during a similar time period of batch manufacturing applications. For these particular products, that translated into an estimated between $100 and $500 million in early revenue for those companies. So a substantial financial benefit at helping it get market a little bit earlier. We looked at other aspects like, were there more post-approval changes for the continuous manufacturing processes, or this impact on pre-approval inspections? We found actually for both of those, there was no really substantial differences between those two types of applications, whether we're talking about continuous manufacturing or batch manufacturing. Yeah, and looking at all these actions together, how did they address the regulatory hurdles that we heard, from industry? So one, early on in this journey, we heard, feedback from industry. We don't think we can implement continuous manufacturing without more engagement from the FDA, right? So, that led to, in part, the creation of the Emerging Technology Program, right, which is a program really focused on that early engagement and collaboration. You don't want to be, the first product approval of CM, right? You want to be that fast second. We hear a lot of that from industry, and then we saw in 2015, that first approved product manufacturing with continuous manufacturing was a big milestone. And since then, we've accepted over 50 proposals from industry concerning, concerning implementing continuous manufacturing into the Emerging Technology Program, and we've seen an additional approval of 15 products that use continuous manufacturing to make those products. So a big switch. Another feedback was, well, we were hesitant to implement until we have guidance from the FDA. So, that has been, FDA has published draft guidance actually in 2019, and for the biotech side, published in 2022 about viral safety, which has, for continuous biotech products, that's a key element for developing those types of processes. Another hurdle is like, well, we don't want to implement continuous manufacturing because we fear it's going to impact our approval timeline, right? I think the self-audit I just talked about showed that actually CM applications were approved faster than similar batch manufacturing, addressing that concern. Another concern is like, well, it's great, FDA is doing all this work, but what about, we produce for the globe and we have to interact with all these other regulators. So, we don't want to, implement CM without international, harmonization. And, this year, actually, the publication of the harmonized guidance of ICHQ 13 really is a milestone in addressing that concern. So I think, we continue to listen to, stakeholders as they are looking to implement these novel technologies that can have, impact for both their company as well as for patients, and trying to be responsive, to that feedback with our programs. And just some of the lessons learned as we went through this journey is that, again, that, we start off this journey with the idea that there could be potential benefits. And I see in a number of cases we've demonstrated, our industry has demonstrated that there are real benefits, maybe not for every product, doesn't make sense, but, a number of these instances, this implementation of continuous manufacturing really delivered actual benefits. Early engagement and collaboration is essential to get to where we are today. And, that's for folks, for us, who's been through the Emerging Technology Program, but really that early engagement collaboration was very key to making the progress we've seen to date. Science needs to lead the way. I think that investment, I talked about that journey starting in 2014, which it's actually not starting then, right? Because a lot of investment in the science and investment in equipment happened actually before then. And that really was really key to enabling the success we've seen today, and the importance of the FDA's Advanced Manufacturing Research Program to support our assessment and policy activities. Finally, it's important to engage, internationally. Pharmaceutical manufacturing is a global endeavor, so it's really important that we don't take that we really engage internationally. And so, I think that's been ICHQ 13 has been really important for that, and it provides a platform to build on, because that guidance has a number of annexes today, but you can see as new modalities get experience with continuous manufacturing, there could be new annexes added to that guidance over time to really grow the impact of that guideline. So that I'd just like to conclude by thanking some of my colleagues who contributed a lot to the material presented today. And finally, you just say thank you for your time and attention, and I look forward to the panel discussion later in the workshop. Thank you.
Hello everyone, my name is Tony Costa, and today I'll be presenting on continuous manufacturing platform for lipid and polymer-based nanoparticle therapeutics. Before I begin, I would like to thank FDA CDER, Small Business and Industry Assistance, for hosting this great event and giving me the opportunity to speak about some of our work on continuous manufacturing. At UConn, we received multiple awards from the FDA, spanning from around 2014 to 2022, and this work was all focused on continuous manufacturing. So back in 2014, this was really more of a proof of concept, understanding some of the basic material attributes, and performing a risk analysis on the various types of nanoparticles, mostly liposomes at the time, and really understanding how to make these nanoparticles using various methods. We selected a method, a solvent injection method, and then we expanded, looked at downstream processing, how to integrate different levels of PAT into the process stream, and then eventually in around the 2021, we actually developed a skid, and this industrial skid was actually brought to URI and used in a clean room setting to evaluate a liposomal doxorubicin formulation. Pharma industry, since inception, has relied on batch processing, and it has been a rather slow process to implement continuous manufacturing, which understandably is due to the strict requirements on drug products and reducing overall risks of product approvals. The 21st Century Cures Act was one initiative that opened the doors for advanced manufacturing technologies. At the FDA, OPQ has been leading the development of international guidelines, where these guidance for industry documents serve as a backbone for establishing frameworks and developing these continuous manufacturing processes. As a high-level overview, batch processing is carried out using multiple unit operations and requires multiple stages of testing and material storage. This type of process is volume dependent and may have difficulty in scaling, whereas a continuous process operates as long as raw material is fed into the system, with finished product being collected on the output. This type of process is time dependent and easier to scale. A continuous process has many potential benefits, for example, providing higher quality drug products, much more easier to develop controlled process streams, a faster to market approach, shaving off potentially months, and supporting end-to-end manufacturing, where we can combine upstream and downstream processes to have a state-of-the-art vaccine or drug product manufacturing platform. Here we show a liposomal nanoparticle, and the liposomal nanoparticle is made up of a lipid bilayer, which typically consists of phosphatidylcholine or other similar type of lipid, and these lipids may be saturated, unsaturated. They can have different hydrocarbon tail lengths. We also bring in cholesterol, which impacts the fluidity of the bilayer, and then some type of either charged lipid or pegylated lipid on the surface. The charged lipid and the pegylated lipid add to the stability of these nanoparticles in solution, or as a colloidal dispersion. Internal aqueous compartment is where an API, for example, a water-soluble API such as doxorubicin, may be encapsulated, and doxorubicin, when in the presence with certain salts, can form a linear crystal, and other API may actually form or load into the lipid bilayer, and also or a combination of the internal aqueous compartment and the lipid bilayer. The typical size range for these particles is around 40 nanometers on the low end, and then can really go beyond 500 nanometers to 1 micron. But for pharmaceutical applications, we're typically around the 100 nanometer diameter mark. Lipid nanoparticles and liposomes are both colloidal dispersions that share similar particle size characteristics and chemical makeup. LNPs include an ionizable lipid that directly interacts with nucleic acid and at certain ratios form a stabilized dispersion. Hybrid LNP liposome particles exist under certain conditions that form two compartments, a conventional aqueous core and a lipid nucleic acid matrix, potentially allowing for nucleic acid to exist in both compartments. A variety of nanoparticles that we produce using our nanoparticle processing system. These include liposomes, LNPs, and polymeric micelles. The bottom images show liposomes loaded with two different API, namely docetaxel and doxorubicin. On the bottom left, docetaxel liposomes form a unique, multifaceted structure. On the bottom right, liposomal doxorubicin forms a spherical particle with a linear doxorubicin crystal, whereas the combination of both API lead to an elongated, non-spherical particle. This nanoparticle processing system can be used as a tool to generate particles of various morphologies under a continuous operation. Now, from the onset of this work, we took a quality by design approach, which is really a risk-based approach to solving any problem. And on the left-hand side, we worked on multiple Ishikawa diagrams, and what these were really used for was to relate material attributes and process parameters to the potential critical quality attributes that we identified. We used guidance for industries to assist in pointing out some of the key factors or areas that we should be more focused on. But ultimately, this allowed us to make multiple lists, and these lists include material attributes and process parameters. For example, on the material attributes, lipid concentration, lipid pKa, head group, ionizable lipid. On the process parameter side, we looked at different flow rates, temperatures, degrees of mixing, and then we looked to look at the functional relationships between the material attributes, process parameters, and how this all impacted the potential quality attributes. On this slide, we have a high-level process flowchart that outlines stages throughout the process stream. Starting on the left, we have an ethanol phase, which contains the pre-dissolved lipid and possibly a lipophilic API. There is also an aqueous 1 phase, which may be a buffer or combination of a buffer plus water-soluble API, such as a nucleic acid or other water-soluble molecule. Nanoparticles are formed via a turbulent jet and then pass into a conditioning module and dilution port. At the dilution port, the material is diluted by an aqueous 2 phase that may be a different buffer than a 1 phase and used to adjust pH and salt concentrations. At the dilution port, we also have a particle size analyzer that measures online particle size. This is very useful as operators can understand in real-time conditions the particle size characteristics of their formulation. Downstream from the dilution port is a multi-stage TFF unit operating in single-pass mode, allowing for solvent reduction and buffer exchange. Another PA tool, designed as a soft sensor, is used to determine lipid concentration coming off the TFF segment. The material then passes to an active loading module, where certain API can be loaded into or onto the nanoparticle. A UV vis spectrometer, also operating as a soft sensor, has been used to determine percent encapsulation in total API. Lastly, a purification stage, or a third TFF, is used to clean up the formulation, potentially reaching 99% encapsulation and providing a bioburden reduction stage. The material is then collected in a 2D bag, ready for further analysis and to proceed to fill finish. In this slide, we show videos of the nanoparticle formation site, or also the turbulent jet, at various flow rates. In each of the 4 sets of videos, the left video has a dye in the ethanol stream, whereas in the right video, we did not include any dye. The 30-second clip we observe here is actually taking place in about 1 second, and the 4 flow rates, ranging from 70 milliliters per minute to 300 milliliters per minute, all show conditions where a stabilized jet is formed. Typically, at the lower 70 milliliter per minute flow rate, larger nanoparticles are formed, and at the higher 300 milliliter per minute flow rate, smaller nanoparticles are formed. In a continuous manufacturing process, process analytical technology is a required tool that provides data in real time and can be used to control and/or monitor a process. The PAT requirements are important to identify as they can impact other controls in the process stream and data collection rates. PAT can be installed inline, online, or at line. Online and inline allow for material to continue downstream post-measurement, whereas at-line measurements require material sampling and subsequent discarding of the material. Some of the PAT tools are developed as soft sensors with predictive algorithms. Having these tools allows operators and feedback control mechanisms to make decisions and meet specifications. Dynamic light scattering to determine particle size, turbidity measurements to relate to nanoparticle concentrations, and UV-Vis spectroscopy to measure drug encapsulation and total API are just some of the sensors that can be used as tools to control a continuous process stream, ultimately providing a robust nanoparticle platform. One of the setups that we've used was a nanoflow sizer. This is a spatially resolved dynamic light scattering system. It's by in-process LSP. It allows you to attach a flow cell onto the process, and you can actually flow at rather high flow rates and very accurately measure particle size. We've been able to measure individual data points in less than 6 seconds. So this really leads to a rapid online analysis. You can see in real time what particles are being formed. Does it meet your specification for particle size, and is it acceptable within a certain concentration range? So it really gives you a much better picture of what you're developing, and not having to wait till the end of a process stream to see if you are actually within your specification. Example, particle size is being monitored in real time. The z-average particle size, which is one measure of the diameter of the particle, along with the Pauli dispersity index, are being measured. Other attributes, such as shear rate and temperature, are also recorded and analyzed. From the particle size graph, the size is very consistent around 92 to 93 nanometers, and the PDI is averaging around 0.05, indicating a rather monodispersed particle population. Having inline-online PAT in a process stream enables scientists and engineers to relate intermediate nanoparticles to the end product, which can lead to a better understanding of the particle behavior throughout all the downstream processing. Here's an example of the online doxorubicin and lipid concentration analysis. So what we did here is we added doxorubicin for the top graph, added doxorubicin, and then you can see the doxorubicin went up to about 95% encapsulation. So soon after the dox was added, we instantly see a 95% encapsulation reading. Then we stopped, continued to run the system after removal of the doxorubicin, and added again at a higher temperature down at a later time point, and then we were even able to get above 95%, almost closer to 96, 97% encapsulation. So that was just simply by adjusting temperatures at the loading site. With a continuous process, we're able to load doxorubicin within seconds, as opposed to a batch process that may actually take multiple hours to heat up a large container and to load all at once, whereas this continuous process is loading small amounts over a period of time and loading a smaller number of nanoparticles over a period of time, but we're able to do so in a very short period of time. Lipid concentration analysis, we're looking at what's coming off the TFF, so we know that we're within the specification, and for this we were targeting around 25 millimolar lipid, and we were definitely within reason of the specification throughout the duration of the run. Now I just want to go into a design of experiment example. So what we did here is we looked at really mostly material attributes and some process parameters. So we looked at post-formation temperature, aqueous phase flow rate, cholesterol percentage, main lipid type, and the aqueous phase salt. So PBS, phosphate buffered saline, phosphate buffer, or sodium chloride. And both the pH values for the PBS and the phosphate buffer were set to 7.45. And then as a response, we're looking at the particle size, polydispersity index, and zeta potential. These HSPC-based liposomes, a predictive model was generated for particle size and an R-squared of 0.89 was obtained for the 270 runs. We compared the datasets using multiple surface plots and also grouped the datasets. One example is shown in the graphs on the left, which is the particle size data plotted against particle size formation, temperature, and flow rate. These examples are grouped based on 30% or 50% cholesterol. As can be seen from these surface plots, it is that the blue surface profile, which represents phosphate buffered saline, has always the largest particles. The red, which is phosphate buffer, and the gray, sodium chloride, surface profiles are similar for their particle size data. Now, we can use this data to help in-process optimization and a control strategy. And you may notice there are other areas of the surface profiles that are somewhat flat, meaning the particle size does not change with temperature or flow rate. These zones can be used to generate particles with a wider tolerance on process parameters. With that said, these types of design of experiments and surface profiles provide a tremendous amount of data, and the continuous processing system we developed can rapidly produce particles for these studies in a short period of time. Design of experiment study, more of an extended study. We included DOPC as part of the main lipid type. DOPC has unsaturated hydrocarbon tails and a low phase transition temperature compared to other lipids. Across all flow rates and temperatures, the particle size for DOPC liposomes was less than 50 nanometers in diameter, which are very small liposomal particles. DMPC, which has 14 carbons on its saturated hydrocarbon chains, DPPC, which has 16 carbons on its saturated hydrocarbon chain, and DSPC, which has 18 carbons on its saturated hydrocarbon chains, all formed much larger particles. DMPC was able to form the largest particles up to about 350 nanometers in diameter, whereas DSPC formed the smallest particles for the saturated hydrocarbon lipids. In the case of DSPC, only smaller particles formed due to its high phase transition and more solid-like bilayer. Accordingly, small formulation adjustments in the number of carbons on a hydrocarbon chain or bond saturation can greatly impact bilayer physical characteristics and the nanoparticle structure, all of which must be fully considered in pharmaceutical development work. The continuous processing system that we developed is also used to generate polymeric micelles consisting of a block copolymer of MPEG BPCl. Curcumin was used as a hydrophobic model drug, and TEM was used to analyze the particle morphology and structure. We can see consistent particle size and no worm-like or elongated structures for these particles in the top left images. Polarized light microscopy, which are the images on the right, was used as a technique to determine the entrapped model drug, as crystallized curcumin is evident when only in water and not present upon loading in the polymeric micelles, which is A versus B. In addition, the bottom left image of the blank micelles, which is a clear solution, the loaded micelles, which is a clear yellow solution, versus the free curcumin, which is a very yellow opaque solution, all indicates full loading into this nanoparticle system. We continued this work with paclitaxel, and the graph on the bottom right shows an in vitro dissolution with first order release for the micellized paclitaxel, and also as compared to Genaxol PM, which is a commercial product. We have a case study on liposomal doxorubicin. This work was performed at URI's Pharmaceutical Development Institute Training Center. We successfully transported the nanoparticle system from Yukon to URI for this work. Here is the specification for liposomal doxorubicin. Our focus was to match the parameters in the specification to our final formulation while using a continuous process. In this manner, we sought to meet the lipid composition, the final lipid concentration, the total amount of doxorubicin, the loading battery, final external buffer, and the particle size. Now looking at the system that we set up in the clean room area, and again, this is our first system that we developed here. So it was brought into a clean room. We did some initial validation on the system, cleanliness, swabs, internal, external, surface testing, the air quality testing in the room. So we went through multiple procedures to understand what we would need for SOPs and just general protocols. And then in addition, we were set up to run for liposomal doxorubicin. So looking at the procedures and overviews that we were able to develop over this time, so we looked at gowning procedures, draft batch records, draft cleaning SOPs. For the cleaning, we loaded the system as a cleaning validation. We loaded the system with the sucrose, and then we removed, and we looked at the total TOC endotoxin levels, different system SOPs that we provided. Also then environmental monitoring and cleaning cycle development, and then also personnel monitoring for the process. On the right hand side, we show a picture of a general recipe. So this is built into the software on the system, and this gives you really full control over all of the process streams, and for example, changing flow rates and running multiple steps. And this gives you, full control over this process. Particle size, so we're definitely in the range that we anticipated. So we had these 3 runs that were performed. Actually, 2 runs were on one day, another run was on a different day. So we were able to form these particles, 87.3 nanometer in diameter. Very nice, very low PDI, 0.03. Encapsulation, a little bit lower than what we were expecting based on our previous runs, around 86%. However, these were run a work in progress. The heating stage was set to a lower value than what we probably needed to run at, so that would have easily brought the temperature up. But we were limited to in certain circumstances for this particular run while the system was at this facility. So in that case, we did get a little bit lower, but 86% is still a very nice encapsulation for a single pass encapsulation. So overall, runs were very successful, and these low standard deviations and percent errors were reported. EM images on this slide demonstrate a clear picture. Continuous processing has the ability to produce more uniform particles when compared to batch processing. The technology developed at UConn on the left have majority spherical and linear doxorubicin crystal structures. The brand product in the center have more elongated and less spherical particles. It can also be seen empty liposomes and possibly free drug and needle-like structures. The generic product on the right, which has a wide range of particle sizes and morphologies, with some large empty particles and needle-like structures present. Inherently, the loading processes are similar, but the timescale changes significantly when a continuous process is used. Being able to load the API in a matter of seconds versus tens of minutes to hours can lead to a more efficient active loading process. Overall, a continuous process provides the tunability required to optimize a process stream. In the continuous process, we can feed this back into models to better understand and predict the process. For example, as shown in the plots on the left, we can determine concentration steps and dilution steps required to meet salt and total drug concentrations. Accordingly, these process parameters can then be fed into a recipe as an optimized condition. On the right, a subset of the overall data is displayed for a typical run, indicating the enormous amount of information we can generate from a continuous process. Next, having the ability to really control the crystal morphology. So this is an interesting aspect that we were not planning when we started off with these with this work. We were looking at liposomal doxorubicin, and most of the time we were making nice linear crystals. But then we started on some formulations to see that we can actually form majority of the crystals as a circular crystal. And on the right hand side, you can clearly see all these circular crystals that are present. And even the ones that actually are that appear to be linear, if we use the cryo-TM tilt at 45 degree, and we're able to actually see that these are not linear crystals, these are actually spherical crystals. When we tilted the images on the left, we could see that these were actually linear crystals and not they also were not spherical. There's a couple spherical crystals in there, however, and you can see one or two present in this image. So that may be another difference between continuous processing, is that you're able to go from linear structure to circular structure of an API within this type of microenvironment of a liposome. So very interesting, how could this potentially impact anti-cancer therapies, and is there a benefit of having a linear versus circular crystal on any potential dissolution or release mechanism? And then, in general, understanding the structures of the liposomal doxorubicin. So you can, and see here the elongated linear nanocrystal on the left. We have more of a spherical linear nanocrystal. So it looks like the crystal size is roughly the same with respect to the number of fibers within the crystal. But then when you look at a spherical circular nanocrystal, you can see how it wraps around and how it forms this very nice circular type of morphology. Then you have some potential U-shaped, so these are going to be circular, or over a longer period of time they connect to form circular. So these are just caught as an intermediate type of nanocrystal. Then all this comes down, how does this impact crystal? How does this crystal structure impact potential clinical adverse events, PK data, etc. As liposomal doxorubicin is to determine the individual particle aspect ratio. This is done by using software such as ImageJ to compare the shortest and longest axes, where a ratio of 1 indicates a perfect sphere, and an aspect ratio greater than 1 indicates the elongated particle, for example, especially above a value of 1.2. From the graph on this slide, the formulation that was developed using a continuous process, which is in red, had all the particles with an aspect ratio of less than 1.15, indicating very spherical populations, whereas Doxil had many particles above an aspect ratio of 1.25 and even out to about 2.0. This indicates much more elongated particles present in that population. And now we also looked at different orthogonal sizing techniques, so cryo-TEM, number distribution, and actually just basically counting the particles and looking at their particle size using an ImageJ approach. Or we have a microfluidic resistive pulse sensing. This is a Spectrodyne NCS1. We tested the particles, and we can, and this is more of a volume distribution. And then looking at the cumulant, DLS intensity-based analysis, and all these, in all three approaches, we were able to see very similar particle size trends. If you get a very low PDI, 0.029, so really anything less than 0.05, we get more uniform particle populations. As we get above 0.05, which most people talk about 0.1 being an indication of monodispersity, but as you get above even 0.05, you start to see potentially that there may be some larger particles or smaller particles mixed in with the formulation, which is picked up more predominantly by a cryo-TM or this volume distribution technique. And again, then going back down to larger particles, 124 nanometers in size, low PDI on DLS. And we can also see, both respective the volume and number distribution techniques. Now, another question is, when looking at these nanoparticles, how it's being measured, by what technique, and even within the same technique, for example, dynamic light scattering, you could have potentially two different light sources So 633 nanometers, that could be a laser source versus just a standard light source. And so looking at a light source of 1300 nanometers, you really do get different results. So this has to go into a control strategy, understanding what system was used, what light source was used, and what are the expectations. In one case here, we see that we get higher PDI using a 1300 nanometer light source. In both these cases, actually, we see higher PDI. And then in this other case, we see, we get a very, very low PDI, extremely monodispersed, 0.008 and 0.039. So, which one is acceptable? Should we accept 0.1 to be acceptable for nanoparticles, 0.2, or 0.05? So really, this helps point us in the right direction of different strategies that we can develop. As a liposomal doxorubicin in vivo study, we were selected by the Nanotechnology Characterization Laboratory at Frederick National Laboratory for Cancer Research, and this was a great opportunity for the university where we were able to work together and provide them some of our nanoparticles and review the results that they were able to generate with their in vivo study. For this study, we looked at two different formulations, namely NCl425 that was designed to have a linear doxorubicin crystal, and NCl426 that was designed to have a circular doxorubicin crystal. Using cryo-TEM, the number, particle size, aspect ratio, and percent linear crystals, we were able to analyze these formulations. Formulation 426 had over 86% linear crystals, while formulation 425 had over 50% of the circular crystals. However, most likely this is much greater, as a tilt was not done on the cryo-TEM, and as we've seen previously, this would have led to a much greater number of circular crystals. Overall, the aspect ratio of both of these formulations were close to 1, which indicates a near spherical particle. So interestingly, when we look at the z-average particle size, and this is what we can use for inline analysis, we're getting exactly what we were picking up around the 87 nanometer mark for the z-average, and very low PDI. So that's a very nice indication that I was using different dilution strategies, 100x versus 1000x, and we don't really see major differences between the two, which is a good indication that dilution is not impacting the overall particle size which is expected for this type of nanoparticle. And then for the NCl26, we see the same. We're getting around 120 nanometer particles, and then also very, very nice low PDI. Formulation stability studies were performed. An example data set from NCl426 is shown here. AF4 separation with inline mols and DLS was performed on these samples, and the shape factor shift was analyzed for 426 with and without plasma. The minimal shape factor shift observed indicates minimal protein binding, consistent with the commercial drug products. And we saw the same results for the NCl425 as well. A toxicity evaluation was performed for the two formulations in comparison to a marketed product. The levels of doxorubicin and doxorubicinol in the heart, liver, and ear tissue were analyzed. Both formulations had comparable values to the manufactured product. Statistically significant differences were found in doxorubicinol concentrations in the heart and liver, indicating slight variations in tissue concentration profiles. The objective of the study was to evaluate potential bioequivalence of NCl425 and NCl426 in comparison to the commercial product Doxil. PK data for the total, unencapsulated doxorubicin, unbound doxorubicin, and doxorubicinol were analyzed. As shown in the above graph, all these profiles were similar and most likely would have similar biologic effect. However, bioequivalence was not fully met. The purpose of the study was not to necessarily meet a bioequivalence, but to understand potential differences due to linear versus circular doxorubicin crystal formulations. Now we look at an mRNA LNP case study where we mostly looked at particle morphology and how that relates to some of the process conditions. So some of the mRNA LNPs that we formed, what we'd like to look at here was how do the different process parameters and processing strategies impact these types of nanoparticles. And one case here is really focusing on the intermediates. So combination of including or leaving these particles in ethanol and also using a low phase transition lipid led to more of a delamination or blob-like appearance on the outer layer. So you can see as it appears that there's this outer bilayer that's coming off or that's associated with the particles. Then when we have low phase transition lipid and we leave them in a high percent of ethanol for a period of time, we can really see the particles swell, they get much larger, and then they start to delaminate, and there's multiple compartments that are provided. However, when you use a higher phase transition lipid, you reduce the amount of ethanol present, you can get much more of a nice solid core, no separation, very stable structure for these LNPs. So all of this can really be helpful in developing and optimizing your nanoparticle formulation. For the mRNA LNP transfection, we looked at just a 28-day study, and we could see that we were able to, in a cell line for K562 chronic myelogenous leukemia cells, we were able to detect a response, and depending on the high versus low concentration of the mRNA, and clearly significant compared to just a controlled PBS and also the naked mRNA. Now, another area that we focused on some of these grants was on the CFD, the computational fluid dynamics, and also on the MD. And we have a very nice paper that was published in Molecular Pharmaceutics on that explains the coarse-grained molecular dynamic simulations of paclitaxel-loaded polymeric micelles. So on the CFD aspect, we looked at the different jet flows, and we did simulations. Really, that adds information that is more difficult to actually measure. We looked at an eddy current analysis, which includes what's called the delta criterion, and what this is looking at more is the vortices rotation that takes place within these jet streams, and we're trying to relate more of an eddy current or vortice that's being formed with some of the material attributes and some of the resulting quality attributes. So, for example, particle size. On the MD side, we wanted to understand more of the basic mechanism of the particle formation. In this case, starting with just individual lipid molecules and water molecules and ethanol molecules, and then going over multiple nanoseconds, so 10 nanoseconds, 100 nanoseconds, 500 nanoseconds, all the way to 1800 nanoseconds to understand really how the particles may form. And in this potential mechanism, we can see the individual particles coming together, and then they form more of a linear plate, and then this linear plate structure then bends inward, and then starts to form a sphere, and it closes up. So this is a nice indication of how potentially the API becomes encapsulated, and also then leading us to understand the general formation process. Lastly, for this work, we looked at shear simulations, so understanding how when mixing, potentially with high degrees of mixing versus a low degree of mixing, and we can see that the shear before shear and after shear particle size was able to be decreased. So this may have some useful impacts, and where we can relate MD to CFD as a new means to really understand a process going from atomic scale all the way up to to more of a bulk, to bulk analysis. Now on the paclitaxel side, we looked at different PEG, PLA, micelle formation, and for paclitaxel, what was interesting here with this work is we're able to see how the paclitaxel enters the micelle and general the time frame really taking place similar to a liposome around the 1800 to 2000 nanoseconds. In this slide, we see the polymeric micelle structures that can actually form. So on the left-hand side, we have a blank spherical structure. Then we have a drug-loaded spherical structure, and then a drug-loaded rod-like structure, and a drug-loaded worm-like structure. So all of these structures can easily form when producing these particles. So even though this may be a thermodynamically stable system, the actual physical mixing process can also impact and have a high degree of influence on these types of structures. We did find that under a higher shear, higher mixing environment, we would form these spherical structures, and then and lower polydispersity index, and then under lower mixing, low shear conditions, we could actually form more of a worm-like structure, and this would be indicated by a higher polydispersity sample on the experimental side. And another aspect of this work is we're able to look at the free energy of the polymeric micelle dissociation. So again, something that's rather maybe difficult to measure, but we can use this MD approach to understand the different potentials along for say pulling a single PEG PLA and pulling a single paclitaxel molecule away from the nanoparticle, and you can see their degrees of the free energy. So even all the way up to about 80-90 kilocalories per mole versus around 40 kilocalories per mole for paclitaxel. So this indicating drug release versus particle dissociation. And lastly, we worked on artificial neural networks for continuous manufacturing. We took in really an abundance of data that we collected over the years. We added in some of the MD, CFD aspects, and we started to look at this as different models. So multiple input, multiple output model, also multiple input, single output model. So MIMO versus MISO for predicting the liposome particle size and PDI, and then we looked at the different training sets and testing stages, and this is where we're able to actually determine, give you a general idea of liposomal particle size. So if you wanted to say, I'm going to increase my aqueous flow rate and my ethanol flow rate and my osmolarity, and I'm going to be switching cholesterol from 20% to 60%, and also my hydrocarbon tail length, this at what temperature, this will give you an idea of the size of the particle that you may generate. So really, again, great tool, understanding and giving you the ability to really generate any particle size. Thank you everyone for your attention, and now I would like to briefly acknowledge all the people that were part of this work. Firstly, the US FDA for providing us with these funding opportunities and for all their support over the years. At UConn, we had a large team of graduate students and postdocs over the years, and this work helped train these students and postdocs for new careers in the exciting and burgeoning nanoparticle field. The PDI Training Center at URI for their support in the operating of our system in a cleanroom setting, and the UMass Chan Med School for the outstanding cryo-TM images. Lastly, to NCL for all their work on the liposomal doxorubicin characterization and in vivo studies.
Thank you for the great presentations. We'll now enter our break until 10:45 a.m. Eastern Time. Please enjoy your break.